Okay, good afternoon. My name is Alfonso Romero Sandoval. I'm Associate Professor and Director of Research of Presbyterian College School of Pharmacy, which is located in Clinton, South Carolina. Today I will talk briefly, hopefully I can actually be in my 17 minutes lot, about the modulation of the immune interactions in the central nervous system in pain conditions. For these we have used multiple approaches, and one of them is the use of nanotechnology to induce some specific genes in the spinal cord, particularly in glial cells, to try to modulate the responses, behavioral responses, and molecular changes in the spinal cord in animals with pain models or pain conditions. So why pain is important? We have focused lately in my laboratory in the transition from acute to chronic pain. The difference of that is basically that acute pain lasts for a few weeks, and chronic pain is defined as the pain that actually lasts more than 3 months. So it's a very, very long disease and condition. Chronic pain is a syndrome by itself. And there are many causes of chronic pain, but one of the probably most important is chronic pain after surgery or post-operative chronic pain. This is due to mostly major surgeries. For example, small surgeries such as appendectomies or hernia repair surgeries. These are minor surgeries, and the percentage of patients develop chronic pain under these conditions is probably 5 to 10%. But in surgeries which involve a lot of tissue damage, like mastectomies, arthroscopies, etc., those patients have 50% chances to develop chronic pain. That means 1 out of 2 patients will develop chronic pain, meaning more than 3 months pain. Most of the time is a couple of years or even indefinite time. This is a very prominent issue because just imagine how many patients are undergoing surgeries right now and in the United States or in the world. So a large proportion of these patients may develop chronic pain. And the problem is that chronic pain produces a large proportion of patients that has disability. So it is a big burden in our economy due to loss of productivity as well, not only in the public health system. But in the public health system, we actually spend more than 48 million in surgeries, due to surgeries per each year. And the public health system economic burden is more than $100 billion per year. This is much more than cancer and diabetes together. The reason is simple, because cancer patients have pain. Diabetic patients also have pain. Post-surgery phenomena involve also pain, etc. So pain is a very common disease in many other conditions. The problem as well is that we don't understand why acute pain transforms to chronic pain. We don't understand. We have some clues, but not really a big idea or a better idea at this moment. That is translated in the lack of proper treatment. Right now, we don't have treatment for these patients, which is devastating, right? Because the amount of patients with chronic pain is large. It's a very debilitating disease, and we don't have treatment for these patients. The best treatments that we have are drugs that are not developed for treatment of pain. They are antidepressants and anticonvulsants. And these drugs provide only 30% relief in 30% of patients. So we still have 70% of the population that has no relief of pain with the current treatments. And the patients that get some relief with these drugs have still 70% of their pain. So obviously there is a big need of new treatments for these conditions. Anyway, after nerve injury, chronic pain is developed. For example, a spinal cord injury or postherpetic neuralgia. This is a direct damage to the nerves, to the nervous system, peripheral or centrally. And this condition in most patients develop chronic pain. This is time, so pain is enhanced. In parallel, we have found that patients, for example, with chronic pain after HIV infection develop an overexpression of MAP kinases in the spinal cord. And this is correlated with chronic pain. In animal models as well, pretty much everything that we do, peripheral injury for transection or ligation or any sort of injury in the nerves, will produce pain-related behaviors in rodents in parallel with an increase of MAP kinases expression in the spinal cord. And if we inhibit these MAP kinases, we resolve pain as well in animal models. In surgical injury, like any type of surgery, any one of us has have a family member or a friend that has been undergone through surgeries. So pain is very strong 1, 2, or 3 days after the surgery, when the patient actually coughs, when they stand up, when the doctor touches the wound. This is painful. This is mechanically evoked pain. It's very painful. But after 1 week or 2 weeks, most of the patients, in general, especially minor surgeries, they're fine. Pain is gone, and they go back to work with no problem. If the damage is extensive, the story is a little different. These patients may develop chronic pain. But in acute postoperative pain models, in animals, also pain goes up and goes down, very similar to what happens in humans. And MAP kinases are overexpressed in the spinal cord and then resolve in parallel with pain. All go together. In chronic postoperative pain, especially in major surgeries, we have this pain that it has to resolve does not resolve, stays chronically. And guess what? As well, MAP kinases stay up. So this is an association, but it could be some causalities there. So what is happening here? What is driving MAP kinases phosphorylation to be deactivated in acute pain that resolves pain? We postulate that whatever is happening here is not working here and is not working here. Whatever happens in acute postoperative pain is not occurring in chronic neuropathic pain or chronic postsurgical pain. So we try to understand, our laboratory is focused on understanding what is driving the resolution of acute pain in order to actually mimic this in chronic pain conditions. Most of the pain research is doing the opposite, trying to understand why this is actually produced, but we don't know what is going on here. Why does acute pain resolve? So we're trying to answer that question. So this is the typical and normal cascade of MAP kinases after injury. Toll-like receptor activation or tissue injury will phosphorylate MAP kinases, mostly ERK, P38, or JNK, and these will actually be the main triggers of pro-inflammatory or pro-analgesic, that means that produces pain, molecules, cytokines, chemokines, or other components, neurotransmitters, that actually are very active in producing neuronal sensitization. Neuronal sensitization in pain is very important, because what happens is that the neurons that feel touch like this doesn't produce pain, because we are activating specifically sensory neurons. But when there is an inflammation, tissue damage, or injury, or nerve injury, this touch now becomes painful. Sunburn, right? That's the typical example. When you put your shirt, it hurts a lot. That's because of neuronal plasticity in the periphery. That is neuronal sensitization. This is what happens after surgery, after nerve injury, etc. So we are trying to understand this. So typically, this cascade of events is regulated by MAP kinases phosphatases. So it's an enzyme that dephosphorylate phosphatases, mostly MKP1 and MKP3. And we postulate that these guys are involved in the resolution of pain, but nobody has tested this. Additionally, we believe that another molecule called CD163 in humans, or ED2 in rats or rodents, will also have some role in reducing this cascade and providing resolution of pain. So we will test this hypothesis. There we go. So we believe that MKP3 or MKP1 will be actually driving the resolution of kinases, MAP kinases phosphorylation, and therefore resolving pain. And we also postulate, well, this is just the animation, if we block MKP3, therefore we will produce the transition from acute to chronic pain. So we will actually test both hypotheses, or either the overexpression of MAP kinases phosphatases or the knockout of MAP kinases phosphatases to provide both phenomena. We also hypothesize that CD163 will play a similar role. All right, so this is the knockout animals. We confirm that these animals don't express MKP3 in the spinal cord compared to the wild type. We didn't observe any compensatory mechanisms in terms of MKP1 expression, which is the other major kinase, phosphatase that regulates MAP kinases. And in the behavior, just to explain, what we test, if you are not a specialist in pain, probably you will need a little extra information for this next graph. What we test is mechanical stimulation. As I said before, When the doctor touches the wound, that's mechanical stimulation. When patients cough, when walked, it hurts, it's mechanically stimulated. So we mechanically stimulate our animals, and typically, in order to produce a painful related response, as we do, we require a high intensity stimulation. Like, this is not painful, but if I actually pinch hard, that is going to be painful, right? If I have inflammation, this is going to be painful. A lower intensity will elicit this pain response. So we did the same with these animals. As you can see here, this is the intensity needed at the baseline, normal conditions, to provide a behavior that is related to pain. The behavior that we measure is the withdrawal reflex. Everybody has these reflexes, even worms, because it's very protective. When we touch something that is really hot, we withdraw our hand. We don't think, right? We don't say, oh, this is really hot, it's going to hurt my arm, it's going to damage my tissue, I will withdraw my hand. No, you don't think, right? You withdraw immediately. That's a reflex that is preserved evolutionary in all species. So the intensity to provide this withdrawal reflex is really high. But after surgery, after a po-incision, very, very minor surgery, it's just an incision in the skin and in the muscles, and then we close. That's it. After that, the stimulus, the intensity of the stimulus necessary to elicit these behaviors is really low. That means that the animals are hypersensitive, right? We interpret this as a painful related behavior, okay? So then, in the knockout animals, what happens? These animals, after 5 days, 7 days, 12 days, they don't recover. They remain hypersensitive. However, in the wild-type animals, these pain-related behaviors return to almost basal levels at day 7 and at day 12. This has been shown before, the wild-type, I mean, by other groups. And this is exactly what happens in humans after a minor surgery. After 1 week, 2 weeks, we return to normal. In knockout animals, we did not observe this behavior. So with this data, we believe that MKP3 is essential for the normal return to basal levels in the nociceptive system. That means that in humans, probably an impairment of these phosphatases can actually be underlying the mechanisms that transform acute to chronic pain. We further did some experiments molecularly. This is the wild type, and this is the knockout animals. In kinases, we observed that p-ERK, and we observed also that p38, the phosphorylated form, is upregulated at day 5, but then it returns to basal levels at day 7 and day 12, both of them, in wild type. However, in knockout animals, logically, because MKP3 is one of the regulators of these kinases, p-ERK and p38 remains upregulated for all the time, again demonstrating that this has an association. Pain and MAP kinases, phosphorylation has an association. So logically, what we did was inhibit this phosphorylation activities at day 12 in these animals. Well, before that, we measured where the kinases were. P38 was found mostly in CD11B positive cells in the spinal cord, in the dorsal horn of the spinal cord. That is microglia. They are mostly expressed there. p-ERK was sometimes we found them in neurons, which is logical. Others have shown this as well. But mostly it was localized in IBA1 cells. IBA1 is a marker for microglia. So p-ERK and p38 are mostly expressed in microglial cells. So we then try to inhibit then the phosphorylation of p-ERK and pp38 in microglia by injecting intrathecally specific inhibitors. And what we found was that this chronic hypersensitivity in knockout animals was reversed by the inhibition of p38 or p-ERK independently. We confirm this inhibition. We have a partial inhibition, we have a partial reversal, and then we confirm that the impairment of MKP3 in spinal cord produces chronification of acute pain by producing an hyperphosphorylation of MAP kinases over time. And we block this. So we confirm that MKP3 is relevant and important for this transition from acute to chronic pain, and therefore for the resolution of acute pain. All right, so then the opposite should be fine. Now we're going to try a model of neuropathic pain. What we did is we transected one little piece of the sciatic nerve, not the whole nerve, just one branch. And this mimics peripheral nerve injury in humans. it produces in the animal 3-4 months of pain-related behaviors. That means that the animals are hypersensitive for a long time. So we know that this model produces pain and also produces an elevation of the phosphorylation of these MAP kinases. So what we want now is to overexpress one of the MKPs. In this case, we overexpress MKP1. If our hypothesis is true, then we will block the phosphorylation of MAP kinases and block the development of chronic pain. For that, we use nanotechnology. We could use viruses, but viruses may have these issues as a therapeutic component. It may have immunogenicities, right? So to avoid that, we use a very novel technology that is nanoparticles that actually wrap our cDNA, our gene of interest, and then we will inject it intrathecally. That means in the space of the spinal cord and the cerebrospinal fluid at the lumbar level. And then we will expect that these nanoparticles will deliver these genes in cells that actually will phagocyte these nanoparticles, in this case microglia, overexpress the gene of interest, and then see what happens. So by using nanotechnology, we were able to induce MKP1. We did observe an increase at the mRNA level, and we also observed an increase at the protein level. These are the Western blots. These are 48 hours after the injection. So we are actually overexpressing MKP1 in the spinal cord of these animals. And interestingly, the animals that were using only the nanoparticle did develop pain at day 1, 3, and 4 after surgery. The animals injected with the nanoparticle plus the empty vector of our cDNA as well developed pain-related behaviors at day 1, 3, and 4. But the animals that were overexpressing MKP1, they did not develop pain-related behaviors at the same intensity that the other models. So with these experiments, we confirmed that the induction of MKP1 at least partially reversed the development, or the development of chronic neuropathic pain. To confirm what was going on, what we did was also measure the P38 phosphorylation level in order to understand if the overexpression of MKP1 was molecularly functional or not. So we did observe a reduction of the phosphorylation of P38 in the spinal cord in our intervention group compared to the other two controls. And then we observed also that this P38 expression was mostly observed in microglia. As you can see, the green is P38. It's a reduction in the MKP1 overexpressed in spinal cord. And also we observed a little reduction of IBA1, but we didn't quantify that. But we did observe a reduction of of PP38. So we confirm now that if we block MKP3, we actually prevent the resolution of acute pain. And if we overexpress MKP1 in the spinal cord, we prevent the development of chronic neuropathic pain. All right, so this is the other molecule that our laboratory has identified as a potential target, ED2. ED2 is the name of CD163 in rodents. CD163 is in humans. We observed the expression of ED2 or CD163 in IBA1 positive cells in the spinal cord. IBA1 is a microglial marker. And then also we observed not a co-localization with GFAP, which is a marker for astrocytes, but the cells are in the vicinity of GFAP astrocytes. Then we learned that CD163 is a marker for perivascular microglia. So because astrocytes are in the perivascular compartment, it is not strange to find CD163 cells together with astrocytes. So CD163 is expressed in a subset of microglia in the spinal cord, particularly in perivascular cells. All right, so this is what we observed. We did L5 nerve transection. We actually did a transection of the nerve similarly to what we did before. These animals develop pain, and what we observed is very difficult to see because there are very few cells in the spinal cord. But this is the sham surgery, so this is not nerve injury, and this is the nerve injury animal. We observe a reduction of CD163 following nerve injury. This was surprising because typically you observe an increase in glial markers after any injury. We did observe the opposite here with CD163 or ED2. Furthermore, we observed that when we inject a CB2 cannabinoid type 2 agonist in the spinal cord, we reverse peripheral nerve injury-induced neuropathic pain behaviors. And in parallel, we observe an increase of ED2 or CD163 in a dose-related manner. I won't show the results of the behavior here. Tomorrow I will talk in my plenary lecture. I will talk about these behavioral results, but we reverse the hypersensitivity. And now when we inject an antagonist of a CB2 agonist, we reverse this effect and we observe again a reduction of CD163. This is just the quantification data. This is a reduction after an injury, and this is a resolution after the agonist administration. And this is the antagonist effect with the CB2 agonist. We, again, observe the resolution. So we have identified as well a potential molecule that is also involved in the resolution of acute pain or in the development of chronic pain. But again, this is just an association, right? We associate this with behaviors, with an agonist and with an antagonist, and everything goes together. But this is not causality. We don't know still if this is causally actually associated to nociceptive behaviors. We did some preliminary data. This is very preliminary. In vitro, we stimulate cells, microglial cells, with LPS, and we observe a reduction of CD163 in microglia. So as we know, LPS is a pro-inflammatory factor, so we observe a reduction of this CD163, again, suggesting the anti-inflammatory or analgesic property of this molecule. When we treat these cells with CP55940, this is a very potent non-selective cannabinoid drug. It acts through CB1 but also through CB2 receptors. We did observe an increase of CD163 or ED2 at 2 different doses. So again, when we administer this drug intrathecally or systemically, we have observed, and other people as well, anti-nociceptive effects or resolution of pain behaviors in animals. So this goes in accordance to these roles of these molecules, of these CD163 molecules. But again, this is just another confirmation of the nature of this molecule, but we haven't yet observed direct causality in this, for this molecule. What we did observe was that when we stimulated cells with LPS, we obviously observed an increased release of nitric oxide, which is a neurotransmitter that is important for neuronal sensitization, and it has pro-nociceptive effects at high concentrations. When we overexpress, when we overexpress CD163, so this is the opposite, we transfect our microglial cells with this gene, this specific gene of CD163, then we observe a reduction of the NO release, suggesting again the anti-inflammatory nature of this molecule. So we hypothesized then, I mean, this is more specific because LPS can actually increase everything, it can reduce everything, and also the CB2 agonist or the CP55 drug, which is a non-selective agonist, can activate a lot of things, can inhibit a lot of things, so we don't know if that was a causality. So this is specifically induction of ED2 gene is also in accordance to the anti-inflammatory nature of this molecule. So we conclude that MKPs, especially MKP3 and MKP1, seems to be important for the resolution of acute pain, and we postulate that an impairment in these phosphatases could be very important for the development of chronic pain following surgeries or nerve injuries. We also conclude that the induction of MKP1 specifically in the spinal cord can reverse the development, or can prevent, the development of chronic pain after surgery, and this could be a potential therapeutic approach for humans. These nanoparticles have been used already in humans with HIV, certainly not in the central nervous system. We are currently doing some experiments in the periphery, which will be more appealing for therapeutic purposes. And then we also confirm that the induction of CD163 results in an anti-inflammatory phenotype in microglial cells, and we postulate that they may be important to reduce chronic pain, as we have seen in animal models. So I would like to just provide a little bit more of reasons to enhance a little bit the debate. This is a neuroimmune conference. So recently, well, not recently, 3 or 4 years ago, actually that is not recently anymore, in the European Journal of Pain and this other journal, I don't remember exactly which journal is this, but p38 inhibitors have been shown to be effective in reducing chronic pain following nerve injury, but also following surgery. So these are small clinical trials. This is very important for us because this is one of the targets for the phosphatases that we are using. So that was very promising. However, this is recently, last year, these guys actually follow up with a larger clinical trial, and actually this compound failed in neuropathic pain conditions. So p38 inhibition by using this molecule does not resolve neuropathic pain. So again, all the animal models have been shown that MAP kinases are involved in pain development, in chronic pain, in acute pain, etc. We all are so excited about it. These are fantastic targets. And once again, in the pain field, we go to clinical trials, large clinical trials, and boom, fail. That's why we have more than 100 years without actually creating a new drug to treat pain. Shame on us, on the pain researchers. Yes. So, yes, that means that all the work that I've been doing is pretty much, I don't know what for, right? So, this is just an alert for you guys. I mean, if you are actually using models of MS, of Alzheimer's disease, of autism, of all this stuff, and everything is beautiful and nice and clear and clean and amazing in animal models, be careful, because in clinical trials, this could be it could be a failure. I will talk a little bit more about the pain and the glial modulators tomorrow in my lecture, and you will see how frustrating is this. Of course, first I'm going to show all the fantastic data that we have, as I did now. But yeah, animal models, glial responses, what is this about? We don't know. Believe me, I've been working in glial cells for the last 8 years or 10. My mentor was one of the huge names in glial and pain, and we have just discovered that the first clinical trial for pain for a specific glial modulator failed 3 years ago. And we've been actually criticized a lot because of that failure. And it's not our fault. We want it to work, right? I mean, it's the story of my former mentor, the scientific career. So anyways, what I was doing with my science, I don't know. Hopefully my postdocs and my students understand that this is science and this is what it is. But anyways, well, this is my funding, NIH, also the Rita Allen Foundation, the American Pain Society. These are the grants that supported these. My students, my postdoc, Christian Dong, Rima Saha, they have moved along. They're in different positions now, but my current students, Candler Page, Laura Bernal, Abigail Alvarado, Bailey Hill, and then my former mentor, Joyce DeLeo, and then Rosa Laundrie as well. Most of these guys are in med schools now, but without these grants and people, this has not been possible. So thank you for them, and thank you for your patience and time. Any questions?